Dear ladies and gentlemen, welcome to the 18th CZELO Science Café. Thank you for joining us. Science Café is jointly organized by the Czech Liaison Office for Research, Development and Innovation, the Czech Centre Brussels, the representation of South Moravian region to the EU, and Mendel University. The series is supported by the Embassy of the Czech Republic to the Kingdom of Belgium. Due to the pandemic, we broadcast online. My name is Pavlína Pancová-Šimková, and I am your host tonight. Science Café is a forum where members of the public can meet scientists to discuss topical issues. It is not a formal lecture or panel debate, but opportunity for everyone to ask questions, express concerns, their opinions about the topic. There is no stupid question, and please have as many questions as possible. Now it's my pleasure to welcome our speaker, Leticia Botella Sánchez from the Phytophthora Research Centre at the Mendel University in Brno. Leticia has developed her career in the field of plant and tree pathology from the molecular to the ecological level. She focuses on the description of novel species of fungal viruses, their occurrence, diversity, and impact. Her alma mater is University of Salamanca. With Leticia and you, we'll mainly focus on the viruses that infect fungi, or microviruses. Novel microviruses can be seen as a potential agents in the protection of forest stands, but also threats for many organisms. Some can be used as biocontrol agents against fungal diseases. Although most microviruses often do not seem to disturb their host fitness, it does not necessarily mean that they are living unrecognized by their host. Microbiology itself is a very young scientific field. The first record of an economic impact of microviruses on fungi was documented in late 1940s. It has also been significantly growing research field in the recent years as detection methods are rapidly improving. But still, it is not as advanced as a human, animal, or even plant virology. Many issues are still unknown. So now I have a first, very fundamental question to Leticia. If we talk about the viruses, are actually viruses alive?
Well, good evening, first of all. Yeah, I think that's it's the key question, no? Because viruses have been traditionally considered something like the threshold of life because they are unable to replicate by themselves. So they don't fulfill the criteria of what we consider a free living organism. This means that they should reproduce by themselves, they should make energy by themselves, they should grow, they should evolve. And viruses don't do this by themselves. They need to infect a cell to do that. So, but the criteria for the description or early life are changing, and actually lately the discovery of huge viruses like giant viruses, which have very big genomes encoding many different genes, and with genomes that are actually bigger than some bacteria, and encoding genes that are included in nutrient intake, included in fermentation processes. These viruses are changing a little bit the idea of what we conceive as alive. Maybe there is not a sharp limit between what's alive and not. Maybe it's just that there is an evolutionary continuum. So some important virologists actually feel very comfortable speaking about viruses as a kind of alive matter and probably the earliest forms of life.
Well, this takes me to my favorite question for this Science Cafe. Because Robert Weiss says that if Charles Darwin would reappear today, he might be surprised to learn that the humans are descended from the viruses the same way as from apes. He stated that in his book HIV and New Viruses. Would you agree with him?
Well, I think that I think that Darwin wouldn't be surprised of this statement, and somehow it is true. We are the consequence of an inherited traits, and many of these traits, considered traits as, for example, genes, are actually given by viruses. Not all viruses, but someone like particular retroviruses that have the capacity to integrate in host genomes. They are actually 50% of our genome, and retroviruses are actually important drivers in our evolution and in evolution of mammalians. I read that actually it's estimated that retroviruses were integrated in mammalian genome more than 500 million years ago. So yeah, somehow that assumption is true, partly true.
So now I understand that we don't fully know what is the interaction between fungi, viruses, and the plants, but how are the microviruses transmitted from the plant to plant or from the organism to the organism?
Well, we don't fully understand the interaction virus fungus plant because it's actually very specific. Each interaction, the outcome of each interaction is specific, and it should be described independently. So, particularly fungal viruses or mycoviruses, they have there are like two theories about their origin. There is one theory, or sorry, hypothesis that points that they have an ancient origin. So somehow the origin is unknown, and they have co-evolved with the host. The point is that they are transmitted intracellularly. And this is a peculiarity of fungal viruses. They never go out of the fungal cell. They are only transmitted intracellularly. So they are very closely related to the fungus. And this transmission can be through the hyphal contact, which is like hyphae are the structures that the fungus have to grow. So when there is a contact between two hyphae that are compatible, the virus can cross. Another way is through the spores, like sporogenesis and or sexual or asexual spores.
So that brings me to another question. If we know and don't know the origin, can they be somehow useful or helpful to actually explain the origin to the disease, meaning that each microvirus is somehow connected to the geographic area, to the fungi, which is typical for a certain region? Can we use them the other way around?
Yes, actually, yes, they can help us to understand the origin of fungal pathogens because, well, it's important to understand that for particularly for forest pathology, that is the field where I work, the bad guys are actually the fungi, not the viruses. So nowadays we are living in a moment where there are a lot of there is like the world trade is enhancing the movement of plants material and from one continent to another. So this is enhancing the appearance of fungal pathogens, invasive species of pathogens. So it is very important to know where these pathogens are coming from, what's the origin of this disease, uh, mm, to try to cut that pathway, no? And we can do this comparing the population, like genetic populations, but we can also do it with the use of viruses because, as I said before, they are only transmitted intracellularly. They are very closely related to the fungal host. So somehow they are transported everywhere where the fungus goes. So not everywhere because it also depends on other factors, but yeah, they go with the fungal host where it goes. where these ones go. So, for example, in the case of Hymenocephalus fraxinus, this is the causal agent of ash dieback, which is a very important disease happening nowadays in Europe. This disease or this fungus is thought to come from Asia, from Eastern Asia, like Japan. And so we could compare the populations of this fungus in Europe and also the virus that the European population host with the Japanese populations, and they have the same viruses. So somehow we are confirming the theory that it comes from there.
Well, with the introduction of the high-through sequencing-based methods, which paved the way to the genomic-based detections of the pathogens, we have some better tools to detect them. But still, how difficult it is to actually find the novel viruses or novel microviruses?
Well, it's easier than 20 years ago. I mean that in the past, the traditional methods for looking for viruses were microscope, like electron microscope or chemically. But for microviruses, we need to use molecular biology, and traditionally we have been detecting the genome of the particles produced by the genome of these viruses. So usually fungal viruses are RNA viruses or double-stranded or single-stranded. So we were extracting double-stranded RNA to somehow estimate the possibility or try to like confirm that we have a potential virus or not in this isolate. But these traditional methodologies were very tedious, and usually it takes you maybe for 12 samples you need 1 day. So it's really like a lot of work, like lab work. So with the appearance, with the advent of new technologies like new generation sequencing and RNA sequencing in particular, we are able to detect many more viruses in many more isolates, fungal isolates at the same time. And actually, we can detect, we go much more deeper and faster than with the traditional methodologies.
You mentioned cholera, and other diseases. We have a question coming to us through the Science Cafe, and it's question, we have a problem in our forest with cholera, how we can protect, or can we actually use microviruses to protect the ash trees?
Well, I think it's important the prevention. That's the most important. So I don't think that there is much to do nowadays because this is it's a kind of pandemic. So it's really the disease is spreading very fast. So it's everywhere not everywhere in Europe. There, I think the front line of the disease is something like middle France or something like that, like central France. And then Italy, also the Apennines, Balkan countries. So I don't think we can do much, but trying to look for resistant trees, trying to look for some biological control possibilities, prevention, like prevention management, like clearing, cuttings, all these things.
Well, I would like to go back to the microviruses or fungal viruses. You mentioned that we have new tools, new techniques. How many of them have scientists actually identified? What is the number? I mean, it's tens, thousands, millions. Where we stand?
You mean like fungal viruses?
Yeah.
I actually don't know the number, but I would say that we are in more than 1,000 for sure. With these new technologies, we are identifying novel viruses every week. And it's very difficult. It's actually very difficult to name all of them. I don't think we are going to count all of them. And I don't think we are going to put a name to all of them. Not only mycoviruses, but it's estimated that we only know 1% of the species, viral species richness on Earth. Like every single organism on Earth, it's a potential host of viruses. There are viruses in the oceans. There are viruses on Earth, in fresh waters, in soils, everywhere. So I don't think that it's easy to put a number because it's updating almost every day.
Okay. When we speak about them, there is another question coming through the chat about the transmission. So is there a possibility that human or animal viruses can survive in the fungal cells and vice versa?
Not as far as I know. Like it's possible that fungal viruses replicate and survive in plant cells, because they have actually many of them can have a common ancestor, and in many like there are many researchers that point that some fungal viruses are very related to plant viruses. So especially regarding viruses that are hosted by fungal pathogens of plants, yes, they can replicate, they can survive in plant cells, but I wouldn't say that in animal cells. It's like really very far away distance. They are very, very far away.
Very far away, so we are safe at that point. But on the other hand, if they are sometimes used as a biological controls or might be used, what is the mechanism? Do they actually stop the fungi from growing? Do they manage the replication of the cell? What's happening when they might be used as a biological control agents?
Well, that's the point. A mycovirus that it's a potential biological agent, a control agent, should decrease the virulence of the fungus. This means that it should decrease the sporulation, it should decrease the growth, and eventually in the tree it should decrease the disease. And this is something that has been demonstrated with one particular fungus, which is Grifonectria parasitica. This is the causal agent of chestnut blight, which was a very important pandemic in Europe and North America. So the presence of a virus, which is called hypovirus, in this fungus decreases the virulence of the fungus. So researchers have been able to demonstrate that also the inoculation of these fungal cells of Grifonectria hosting the virus are able to spread naturally to other, let's say, non-hosting virus cells. So in the moment that the virus is spread, it's transferred to another fungus, these other fungus start to decrease the virulence, and eventually in forest, in the tree, the disease start to attenuate.
Okay, now I have a slightly different corner to ask, because while studying for the Science Cafe and for the debate with you, I came across across your recent article. I think that's the first one after your parental leave. Well, it says it's called Marine Oomycetes of the genus Phytophthora Research related to the buina viruses. That's the title. Well, what is the article about? What's the story?
Yeah, well, Phytophthora are oomycetes. They are not exactly fungi. Oomycetes are more related to diatoms and brown algae. And in particular, Phytophthora, they are marine oomycetes. So they have been traditionally considered like decomposers and not dangerous for plants. But lastly, they are actually pointed as maybe they are not so harmless. So because there's some study that point that they can be dangerous, and they can be dangerous for some seagrasses. So when we got a big project about Phytophthora, we were thinking about where to start, and we decided to start with Phytophthora because we were wondering, well, maybe this change in the behavior of these oomycetes, it's maybe because there is a virus there. Because viruses actually, they can decrease, but they can also increase the virulence sometimes. It's kind of odd, but it can also happen. So we were looking for possible viruses in a collection of isolates of Phytophthora, and we found out like quite high diversity of actually single-stranded RNA viruses, negative ones, like negative single-stranded RNA viruses, buina viruses. So these viruses are present. The variability is high. They are only transmitted between the same species of Phytophthora. And it's a point of starting point for other works, like what's the role of the viruses there.
We have another question coming through the chat. So the question is, if a virus can be only identified via sequencing, will it be ever described as a new virus, or it is necessary to isolate them first?
It's not necessary, for the description of a novel virus, you don't need to isolate the particle. Nowadays, the species description is based on phylogenetic analysis and a comparison of the sequences, like nucleotide and particularly the protein sequences.
Okay. So,
but of course it helps, and actually it's also very good if you are able to check how it looks like under the microscope.
Yeah, there is another one. Is there some way that a fungus can heal itself from the virus? Or is it like a therapy against microviruses? Can fungi actually push out the virus?
Yeah, actually somehow yes. And yes, because fungi, they also have their defense systems. So they have let's say there are two known ways. One is the vegetative compatibility, or something called itself recognition system. So when a fungus doesn't recognize another fungus as compatible, it creates a barrier that stop the virus transfer or any other possible harmful agent to this fungus. So that's a way to do it. And this is actually a very important point for the use of viruses as biocontrol agents, because if the if the fungal host has a lot of compatibility groups, it can dramatically decrease the efficiency of the transmission of these viruses.
So I hear that
sorry, I would like to say that also at cellular level, this is let's say that this is at population level. At cellular level, fungi have a small, it's called our interference RNA system. So it's something like a system that recognizes a virus as an invasive agent in the cytoplasm and try to block it and break it into pieces and dismiss it. So they have their own defense systems.
So, as I understand, there are many, many questions. So my next question is, which one you are the most eager to answer? What is the one that you want to? What do you want to know?
Well, in my case, I really would like to go deeper in the interactions that I'm studying, because I'm working in the field of forest pathology. So usually, traditionally, it's more focused on answering faster questions. That's actually it. I mean, I agree with that. It's foresters need faster answers, but I would like to answer questions about these interactions. I mean, deeply, like a transcriptional level, a proteomical level, and in particular with some important forest pathogens like Phytophthora cinnamomi or Dotistroma.
Well, you lived and work in Spain, Finland, and the Czech Republic. This might be related. What is the positive side of the researcher's mobility?
Well, I think it has many positive sides. I think it's actually crucial to move, like to go to other laboratories, because it's a way to learn new techniques, to learn how to work in different environments. Also, to realize that what you had in your previous lab was actually not so bad. It's actually there are good things everywhere. I don't know. It's like it gives you a much more capacity of adaptation, and I think that's crucial. I think that's also very motivating, actually.
There is another question which is related to it and coming through the chat and what are the conditions of work for a scientist at the university in Czechia?
Well, I have to say that I've been lucky because I when I was looking for a postdoc, I was living in Finland and preparing a call. But then I got to know about the possible postdoc position in the group where I'm working now, and I have to say that I've been lucky because of I have felt very supported by my department, the department where I'm working. But if I see around me, I think that there are a lot of possible improvements. And I would suggest several ones. I think that, for example, it would be important that PhD students, they don't get only a scholarship, but they have some recognition, like real workers, because they are doing a professional scientific work that it's going to be published. So I think they should not only have a scholarship, but some kind of contract. And I think that the system is more dynamic than in other countries, and you still have the possibility to have a long term career. So I don't think it's so bad. It could be better, but it's okay.
So this brings me to maybe a related question. How difficult is it actually for a woman to have a career in science?
Well, I think it's as difficult as for anybody up to maternity. From maternity, everything changes because you somehow stop, no? And when you stop, like to be a scientist is something like a lifestyle. So it's not like you stop being a scientist for 1 2 years. It's like if you are a sport person, if you stop running for 2 years, it's difficult to come back to competitions and to be at the same level as your colleagues. So that's the most challenging part, uh, honestly.
Well, microviruses is not exactly a typical thing to study. So the other thing that I would like to know is why did you actually choose this field or this discipline? And microbiology is seems to be quite young discipline. So why they are unique to you?
Well, I started working on a fungal pathogen called Gremmeniella avietina. It's a conifer pathogen, and then little by little, when you start to do your PhD, you get much more you close the field little by little, and at the end, you are working on something very specific. So it's something like that happened to me, that I started studying the populations of this pathogen in Europe, and then the Finnish group where I was working with, they were studying the mycoviruses. So little by little, I got I hooked on mycoviruses very much, and I like working on them, maybe because I don't know, I like their simplicity and their potential, and the potential consequences of their presence in trees, in something so big as a tree.
Okay, so now at the European level, we are so much debating about the open science and especially open data. What's your opinion about that? I mean, for the genomic sequencing, there is enormous number of the data which is produced. So what's your opinion about this open science and open data approach?
Well, I do agree with open data as far as it respects the authorship, because we generate data based on projects that we work on and we applied for, and that's like a lot of work to do. So yeah, I think that this is for me that's important. Like as far as you respect the authorship, like the ownership of the data, it should be open.
We have another question coming from through the chat. I do apologize in case I don't write or if I don't read it correctly. And please try to answer it as we all laymen would be also able to understand. It is the goal in nowadays microbiology to achieve something similar with other pathogens as was done with the CH1 in cryptomere, or it is hardly expected?
No, there are actually other examples. I mean, it's true that the example of the Cryphonectar hypovirus one, it's very unique, and it's so far the only successful example of the use of mycoviruses as biological control in the field. But there are other very interesting examples in other fungi like Sclerotinia sclerotionium or Fusarium oxyosporum. There are other examples, and actually, in the last years, this small field of research it's expanding, and there are more and more groups studying different fungi. So I think that there will be successful examples in near future, like applied in the field.
Okay, I have another question coming again from the different corner, slightly related to the situation at the Czech universities. Is it easy to understand to the Czech society?
Yeah, I see Czech society as a European society with its own idiosyncrasy. But yeah, I think that it's a modern society and more European now than in 8 years ago when I arrived. And yeah, generally yes, I think yes.
Leticia, what keeps your motivation? Yes, I mean you to deal with the tiny microviruses and to deal with the being in the different country. What keeps you motivated to actually focus on your science?
Um, I think that especially cooperation with other researchers, congresses, scientific discussions, internships, citations of your work. Like to know that what you do is interesting for others, and somehow you are helping to understand something bigger. That's what keeps my motivation. Also, some small successes in the lab. I don't know. Like a positive PCR or a negative PCR. Like small results keeps you motivated.
When we speak about the current situation, does the pandemic somehow influence your work?
Totally, totally, because well, I mean, I have a lab work to do. I also have like computer work to do. Like a researcher is always doing something in the labs or something in the field or writing some manuscript or writing some project. So if the university if we are under lockdown, it means that I have to work from home. It means that I cannot do the lab work, so I have to focus on other things. But it's quite challenging when you have kids at home.
Well, then I have another question
And
related
also,
to.
just to say, sorry, that we don't have meetings anymore. We don't have congresses anymore. I mean, it's great that we can use online tools to discuss, but we don't have these meetings anymore. And I think yeah, it's very sad.
So the personal contact is necessary for the science and for the scientist.
I
How
think
do you, well, on the other hand, I know that your husband is also a scientist at the Mendel University. How do you keep your work-life balance? Is it not turning to the way when it's under the lockdown that the debate is about the samples and the PCRs and something else? Or how do you distinguish between the work and home time?
I don't think we distinguish it. I mean, it's not divided. I think it's actually what keeps our life more balanced, that we understand each other. So we work in the same university, and actually, I don't know, we have enough flexibility. I have to say that we have enough flexibility to manage our situation. So
Okay. On that note, where is the future of the microbiology? Where do you see the future of your research? I mean, where is the field going? Which direction?
yeah, I think that the metatranscriptomic studies, metagenomic studies are really like nowadays what is expanding our knowledge, and it's gonna help to understand to have like a much more complete picture of what are microviruses, where they come from, and what we can do with them.
And do you think that we will be actually able to do anything with them in the close future, or is it something that we are expecting in long run?
No, I think that there will be, there are some examples, like there are some current researchers that are studying particular models of fungal, like viruses and fungi, fungal pathogens, and I think that in not very long, let's say in not short but not long term, they can be applied in field, and they will be successful at least for a while.
So if I turn it the other way around, do you think that the plants are actually under constant attack by fungi, by some other things?
Well, they are like, but it doesn't mean that they develop a disease. I mean, it's very important to understand that also plants have their own defense systems, and I think it's important to understand that that most of the pathogens, not only viruses, but most of the pathogens that coexist with a plant in a particular environment somehow they have co-evolved. So a pathogen is continuously attacking some particular species of plant, but this plant has also learned to defend itself against this particular pathogen, a fungus or a virus or whatever. Uh, so there is an equilibrium. The problem comes when a pathogen jump into a different ecosystem, and it's able to attack a particular plant that didn't have enough time to learn how to defend against that pathogen. And this is what is happening nowadays. Also, um, stress is something that somehow predispose plants to the attack of of um, of pathogens, so predispose their health state, so it weakens plants. So acidity, higher temperature, water stress, all these things that are happening in many different forests, in many different ecosystems, also predispose a plant or a tree, um, and make it weaker and more susceptible to be attacked.
We have another interesting question coming through the chat. What would be the next country where you would like to do your research? What's the next destination?
Well, I would like to learn a particular technique that a group from Japan has applied for many years. So hopefully, if COVID let us travel somewhere and my husband agrees, we will go to Japan.
So the next destination is Japan.
Hopefully, I don't know. Of course, I also would like to. I have different colleagues in different places, and they are doing very interesting things. But I usually decide where to go based on my family and also on the techniques I want to learn. And yeah, let's say that maybe Japan.
Do you think it's easy for the scientists to actually move around the globe and around Europe,
Yeah, I think so.
even with the family?
I don't think it should be it's such a problem. I mean that you don't need much. You just move somewhere and I mean I don't want to move forever. I'm thinking about an internship of a couple of months. I'm kind of established in Czech Republic.
Okay, so at least for now, it's the Czech Republic, your scientific destination.
I hope so. I hope so that we have enough projects, we have enough money, and we can continue and have stability.
Well, with that positive note, I would like to all thank to you and to all our participants for joining us.
Yeah, thank you
And
very much for inviting.
yeah, it was a pleasure to have you. And stay tuned for next Science Cafe.